Hello everyone. I, Dr. Pooja Chawla, welcome you all on behalf of entire ISFCP family. Under the aegis of IQAC-ISF series, we are organizing the dialogue series, and today the expert for today's talk is Dr. Sapna Malviya. I take the opportunity to introduce Dr. Sapna Malviya. Dr. Malviya is currently she is the head of the institute, Modern Institute of Pharmaceutical Sciences, Indore. She is also president of Women Forum SPSR. Dr. Malviya is also chief mentor and advisor MSME sponsored incubator, innovation ambassador IIC, MOE's incubator cell. Professor Malviya has attained grants of more than 30 lakhs from All India Council for Technical Education, Madhya Pradesh Council of Science and Technology, Bhopal, MPCST, Atomic Energy Regulatory, Mumbai, and Entrepreneurship Development Institute of India, and Ahmedabad in the field of pharmaceutical sciences. She has also received support from IIC MHRD for coordinating impact lectures. She is Innovation Ambassador, MOE Innovation Cell, Government of India, and Institution Innovation Council, Ministry of Education Initiative, Chief Mentor and Advisor of MSME Sponsored Incubator. She has received her pharmaceutical education from B.R. Nahta College of Pharmacy, Mandsaur. She is qualified ZED consultant to pharmaceutical industries. An academician par excellence, she has superlative research expertise in pharmaceutical sciences. She completed her B Pharm and M Pharm. She added another postgraduate degree in management. Later, she finally completed doctorate in pharmaceutical sciences. And as I have mentioned, she is currently working as head of the Institute of Pharmacy at Modern Institute of Pharmaceutical Sciences, Indore. So, without taking much time, I would like to welcome Dr. Sapna Malviya for her lecture. And the topic of her talk today is Drug Discovery and Development from Medicinal Plant, Modern Era of Separation of Plant Metabolites, utilizing advanced AI integrated techniques. So, thank you so much, Dr. Malviya, for accepting our invitation and sparing your precious time for us. Over to you, ma'am.
Okay. So thank you very much for the kind words, ma'am, and invitation. I am heartful thankful to GD Gupta sir for giving me this opportunity to have an interaction with you people. So starting with the session, why I have selected this topic: drug discovery and development from medicinal plant, modern era of separation of plant metabolites using AI integrated techniques. See, basically. For in drug discovery for the medicinal plant, only the important thing is that right kind of chemical constituent in its development stage. Now herbal industry has developed a cumulative profiling and protocols for various type of products which are there in the plants, and which at the right time of their collection, so that we can get in right chemical constituents as well as right type of approach for that particular chemical constituents. See, every one of you know that phytochemicals are rich in the plant, and they are very beneficial for the disease and the long term health management of the patients. So we are moving towards the plants, and plants are consumed at the every part of India, because they majorly consist of various chemical constituents. Say, if I'll name the few: saccharides, coumarin, flavonoids, they terpenoids, steroids, they are working for majorly the health benefits. So these compositions are very important. Now, drug discovery. Drug discovery is a leading challenge. If you'll talk about, it's a leading challenge scientific task, and it's a robust technique. It's challenging because its screening procedure involve a new to find out the new isolate, which is screened at the level to get an expertise of expert because of the chemical structural diversity of this plant, so that they can be employed for the development of new products which can be screened at the different level for discovering new drugs. So we are in the trend of developing these products with the use of artificial intelligence. Now, drug discovery with the natural plant is a challenging task for developing a new leads. Why? Because this particular investigation require discovering new and effective drug components, and these leads to an viable candidates, and you will require various type of sophisticated screening of the products by using GC, MS, NMR, IR. Every type of cumulative profiling is there, because every time we will not get the single constituents which can be evaluated easily by the sophisticated instrument. They are the multiple variant resources, and these multiple variant resources can be evaluated by different procedures by cumulative profiling using sophisticated instrument. Now, why AI? See, we are moving towards industry 4.0 in the fast revolutionary aspect. These revolutionary aspect is being accepted by various pharmaceutical industry, because we need error free operation, so we are going for AI. Now, I already told in the starting of the session that if we are talking about any medical constituent, any herbal plant, first and foremost important thing is that chemical constituent. Now we are in the approach of finding the chemical constituent from the
हेलो
plants.
एक्सक्यूज मी मैडम
Yeah,
मैडम स्लाइड मूव नहीं कर रही प्लीज
sir,
चेंज स्लाइड
I'll move, because I'm
ओके
just
ओके ओके
going with
ओके
the introductory part, sir. So I'll move as per my.
कंटिन्यू
I'll move, sir.
कंटिन्यू प्लीज
I'll move, sir. So we were talking about the AI. So whenever we are talking about the herbal plants, only the thing which is very important is the chemical structure of that particular plant. So chemical structure, whether that particular plant have that sort of chemical structure which has been developed to an extent where we require the extraction. So we are in the stage of artificial intelligence where we can find out the bioprofiling, where we can find out the growth and yield of the chemical constituent in the intact plant. So it is very important, because we know that whenever we talk about the herbal medicines, its temperature, its pH, it's the method of extraction, time of extraction, development stage, everything is very important for select various other parameters like selection of solvent. So here the AI is playing a major role. Now, I told you that we are moving in the revolution where three terms are very important: automation, intelligent, and smart computing. Now, these three terms are playing an integrated role for the development of these medicines and drug discovery and development, to the extent where you can get the lead compound for further discovery of the plant. So whenever we are talking about any procedure which is related to AI, we are getting an errorless procedure, because we are evaluating procedure at every step. So I was talking about automation. Automation means we'll have a completely automated procedure where we can have a live response or live evaluation of each and every step, so that we can get in data, which data can be utilized in the library of the computers to get the intact information. So, since I told you that whenever we talk about any plant or any development of the plant product, the important thing is about the temperature, growth profiling, pH. These all things will be stored in the libraries, wherein people can evaluate through that libraries when to take out the plant, for extraction, so that we can get maximum yield with a good number of primary as well as secondary metabolites. So moving to the next slide. So now you're able to see my next slide?
Yes, ma'am.
Yeah, thank you very much, madam. So I am talking about this artificial intelligence, where it is helping us in every aspects, like soil management, seed sowing, fertilizer and pest management, irrigation management, harvesting, post harvesting technique, marketing management. Everywhere it is approaching the smart learning, so that we can modify our metabolic pathway to such an extent to get a best possible output with minimal input. Now this artificial intelligence is maximizing the yield to an extent to get in bio-based industrial products. So this was the overall theme of the lecture, wherein we will discuss the various traditional, classical approach and modernized approach where artificial intelligence can be implemented and where the basic knowledge is required for starting a successful completion of natural drug discovery. Just giving you the outlook that where to start with the natural discovery. First thing is we should importantly know that there's two type of pharmacy: green pharmacy and red pharmacy. So telling about the overview of the green pharmacy was founded in 1993 by Sain sir. He was a deep believer of the Ayurveda. So this particular pharmacy will have less side effects or ill toxic effects and better formulations for the people. And tremendous number of research is continuously performed in on the literature which is available in the Ayurveda for the selection of the indigenous herb, study of the modern formulations, and how to produce a best product so that overall crowd can be benefited to rediscover the natural secrets and high quality product. Red pharmacy, we all are using synthetic medicines for the disease management, and wherein we have certain sort of side effects also. Therefore people are trying to move towards the green pharmacy, and to move towards the green pharmacy, it is very important to understand the drug discovery and development approach. Talking about the traditional management or traditional medicines, I just want to make a key role of understanding why it is a preferred sort of healthcare, because we people are moving towards this medicine because number of reasons are there. You can say that affordability is there, accessibility is there, cost is also one of the reason. So we in India, natural products are the backbone for traditional medicines. Throughout the globe, people are moving towards because of the herbal medicines. So it is very important role they are playing for this particular development of synthetic, semi-synthetic medicines, and newer approaches are continuously developing because of the impact of the natural drugs throughout India and across the globe. Phytomedicines, which are obtained from the natural medicines to cure the human crowd, they are very important since life immortal. Now I'm coming to the very important aspect wherein we can discuss about the extraction of various products. So because we are in the need of all sort of phytomedicines. These phytomedicines will they are very important because they are affecting the various sort of disorders. We are moving to the era of nutraceuticals. See, nutraceuticals are the specifically designed formulations which are fulfilling the dietary requirements, and they are preventing the they are preventing healthcare also. And these formulations have certain sort of nutrients and various compositions which are really helpful, and nutraceuticals are providing nutritions with pharmaceuticals. So if we need to understand, if we need to isolate, if we need to extract these all medicinal product, so I'm just telling you where on these have the application. Nutraceuticals, cosmeceuticals, these are very important aspect and accepted worldwide throughout the India, and wherein the herbals are playing a tremendous role. So coming to any sort of herbal drug isolation or developing a lead from a natural drug, first and foremost thing is extraction, which is the first step in the phytochemistry. Because whenever you are going for extraction, its objective is to get down the chemical constituent of utmost satisfaction to an extent you will not you're not supposed to get an unwanted material. Means you need to remove all the unwanted material, and you need to specifically focus on the material of your interest. So various type of separation procedures have been given in traditional approach also. By this diagram you can see that how, what is the principle behind to get an extract. Means it is a plant develop extract is there. You will get the molecule, you will select the molecule of your interest. After selecting the molecule of the interest, after collecting and after identification of that plant, authentication is important is there. Then you are extracting your compound by using polar solvent or non-polar solvent, depending on the principle, like dissolves like. And now then you evaluate your plant extract, you judge it, you'll go with the chromatographic analysis, and you will go with the structural elucidation, and all sort of experiments can be done through understanding the particular parameter by which the extraction is really important. So here in the diagram you can see that you are using different type of solvent. It can be polar solvent depending on the like solubility like, and then you will have solid liquid separation, and you will get the extract of your choice by using this method. These are the various technique of extraction which are really important to understand before going to the various type of terminology like means developing a lead component or developing a product component. It is very important, starting with the immersion method. See, basically what happened in the immersion method, you will just take the powder and powder will be put in the extract. Sometimes you will shake the product, or sometimes you will not shake the product, and you will get the product of your choice, of your interest by using this particular method. So various type of parameters are there, like you can go with immersion, you can go with the percolation, you can go with the decoction. Every parameter have its importance. First important thing is that before starting any extraction method, important thing is selection of solvent. Always by selection of solvent you go for non-polar to polar. It is very basic aspect, but you always select it from non-polar to polar. Say if you will start with the pet ether, then you will have different type of solvents like diether, toluene, and then with you will go with methanol, ethanol, and water. Now, all sort of hydrophilic constituents, chemical constituents, are better to extract with the hydrophilic solvents. And other which are lipid loving will go with the lipid type of solvent. So like dissolve like is the basic principle wherein you can understand the basic concept of dissolving the solvent with an extend. Now you have different type of methods. You have soxalate extraction apparatus also. I'll just show you the screen for the soxalate extraction. Now these are the basic approach, like where you go with the laboratory aspect, where you can separate different type of chemical constituent depending on its solubility. Now, plant compounds like terpenoids, steroids possess a low polarity. They can be dissolved with the lipophilic solvents like in chloroform or ether, which I already told you. While the chemical constituents like carbohydrates, amino acids, they have rather high polarity, so they can be dissolved in water, ethanol. So, solvent commonly used extracting the chemicals solvent are based on its polarity. Now you have two type of waters are also there. See, normally we have the mindset that water can be used because it is a cheap, you can easily get the water, so water can be used with the because of the strong polarity. But every solvent had its pros and cons. Same goes with water, although it is easily availability, but the thing that it is very difficult to store because of it easily go to motility or microbial growth is easily there. Additionally, means water has high boiling point, so water extract need to be concentrated for long time. But then if you need to go for a specific parameter, you can use acid or alkaline water, because it sometimes increase the solubility. Like acid water could extract alkaline material such as alkaloids very easily by the formation of salts. Similarly, if you go with organic acids, anthraquinones, flavonoids, these are the compound which can be extracted with the formation of the salt. Furthermore, if you move towards other type of solvents, you can use ethanol also, methanol also. Ethanol is most commonly accepted because of the various sort of the acceptability, because it does not really easily get the microbial growth, and it is accepted because of the strong polarity aspect, and hydrophilic organic solvents such like ethanols are really accepted. Now coming to path ether, benzene, chloroform, ether, ethyl acetate and chloroform, these are other solvent which are not miscible with the water, and they can be applied to extract lipophilic component like volatile oils, chlorophylls. So basically when we talk about the solvent selection, it is a very important parameter for extracting any sort of solvent and before deciding the any type of method. Okay, now we are talking about this particular constant reflexing method which you can able to see on the screen, which is oxalate extraction. So it is also known as constant reflecting extractor, and this apparatus is have many advantage and disadvantage too. If you are really means interested to get major yield out of it, you can go with this particular apparatus, but then it consume too much solvent and it is little bit complex compared to other reflexing methods, percolation and maceration. So it is a method of choice as per the extraction, which type of chemical constituent is there, whether it is hydrophobic or hydrolipic, you can select this particular method. Okay, now means various type of methods are there. Coming to this supercritical fluid extraction method, this particular method can be used by controlling different temperatures and pressure, because supercritical fluid, this particular fluid is used, it is a critical point of a pure substance where highest temperature at which highest temperature and pressure you will get the compound in vapor and liquid equilibrium. So temperature and pressure above this particular point, it's a single homogeneous fluid is formed, which is known as supercritical fluid. So this particular liquid is quite heavy, it has low viscosity like gas, and it has means it has high diffusion coefficient, and it is very efficient method in extracting various compounds and various type of supercritical fluids like ammonia is there, ethane is there, dichloromethane is there, heptane is there. Means most accepted supercritical fluid is carbon dioxide due to its various property, its critical temperature, and its at room temperature it has critical pressure, so which is not too much high, and it is also non-toxic in nature. Various type of parameter, it is odorless, it is non-inflammable, it has chemical stability. So due to low cost, it is accepted among the crowd. So it is basically used for supercritical fluid extraction. It has various advantages because it near to new temperature you can get various active constituents, and the product which you will receive out of it will be of high purity and high yield. It is widely accepted among the various crowd and various advantages, so we go for supercritical fluid extraction. Other type of novel technology where people are using is ultrasound assisted extraction. So this particular method is using ultrasonic waves. It is type of elastic mechanical vibration somewhere around 20 kHz in electric medium, and it could electric medium. So this particular wave vibrate the liquid medium, and Due to this vibration, small holes are formed in the medium, and due to the small holes, the solvent can incorporate in the particular plant material, and whole number of chemical constituent can come out, taking the various parameter of extraction. This particular extraction method has been used. Now, microwave assisted extraction technique, which is also a very important technique, using the microwaves in the different range of different wavelengths from 0.1 to 100 centimeter, having different type of microwave frequency, somewhere around 300 to 3000 Zats, and it is using infrared and microwave ray. Now what happens, what is the principle behind the microwave? So polar compound, basically they absorb microwave energy, and they release energy in the form of thermal energy, which make temperature inside the medium to rise to an extent, and the pressure also increases, so compound flow out and dissolve in the solvent. So this is the basic principle, where the electromagnetic field is also generated to dissolve it and get the compound of your interest. Similarly, pulsed electric interaction is also used. We are using it for extraction, using non-thermal fluid processing technique using high voltage. So these are all method using different type of energies to get out the chemical constituent of our interest. Every process have its pros and cons, and depending on the availability, you can use the different procedure. Coming next to it is energy assisted extraction. So sorry, enzyme assisted extraction. So enzyme, basically plant materials are treated with various enzymes. Various enzymes are there, like pectinase is there, protease is there, cellulase is there, hemicellulose is there. These particular enzyme hydrolyze the cell wall, and they release the phytochemical which bind to lipoprotein or carbohydrate chain to get it out. So this is a pre-treatment approaches by which this particular chemical constituent can go in the solvent. Similarly, steam distillation is one of the methods, sublimation is method, this pressing is the method. Coming to the sublimation, so sublimation, steam directly without melting after heating directly affect the portion of the plant, and it condense into the solid, and after cooling it deposit it in the intact material, and you can collect your chemical compound of your interest. For an example, you can understand of the camphor. You can use camphor, extraction of camphor from camphor wood. Even we can do extraction of caffeine, which is a muroxide alkaloid, by the tea, by using sublimation method. Similarly, pressing also, you can get the desired compound of your interest. Now means after extraction, for the development of any sort of chemical compound, important thing is isolation. Now we are talking about the advanced technique, so we have separated this with the classical isolation method and modern separation technique. Separation of any chemical constituent by the process of isolating the constituent from the plant extract by getting the compound is the next important step before purifying it to a monomer compound. Okay, so finally in extracts you have the blend of constituent, but whenever we talk about the isolate, you'll get a single chemical constituents out of it majorly. Sometime it may not happen, therefore you need to further evaluate, purify, or screen your procedure by various quantifying or bioprofiling, like you can do with TLC, you can go with HPLC, HPTLC, and so on. Okay, so we are starting with the solvent extraction. Solvent extraction method for classification by using a solvent method. Solvent method, you are using acid and base solvent method. Okay, so we are carrying it with different acidity and alkalinity compound. Okay, as I already discussed with you, water soluble alkaline organic compounds, like you can say alkaloids are one of them, can react with inorganic acids to form a salts. And now these salts can be easily separated. Similarly goes with the acid compounds also. Acid compounds, say if you can say carboxyl or phenolic hydroxyl groups are there which are attached. They can be salted by bases and they can be dissolved in the water to get it out separated as an individual components. So these particular methods can be employed depending on the pH value. You adjust the pH to get the acid or alkaline fraction from different type of components, and you can separate it by using different type of solvents also. So again, here the concept of polarity or separation exists is that I already told you that polarity plays a very important role. You can separate the component on the basis of like dissolve like. For an example, I'll tell you that if you are going for strongly polar compound like N-butanol is there, okay, N-butanol water system is there. So these compound have strong polarity. Okay, compound like medium polarity are like you can say that ethyl acetate water system is there, and compound with weak polarity are separated with chloroform ether water system. Okay, so you can decide separation of the material, separation of the solvent, depending on the various principles which we have already discussed. So what happens basically in this operation, that plant extract are dissolved in water first. After dissolving in the water, the solution or suspension are extracted by using the separating funnel with different type of organic solvent. This is a mediocre step. What happens in extraction में क्या होता है कि you have the blend of chemical constituent, and if you directly go for isolation, अगर आप directly isolation पे जाएंगे तो column chromatography will take many hours to separate, because it consists of various chemical constituents which are numerous in number. You cannot count the number, because if you go with the extraction with the TLC, you'll get number of number of separation or number of spots at different RF. So हमारे पास जो बीच का procedure है, that is getting the fractions in the between. These fractions are they can be individual or they can be multi-component, but still they are the purified form of extraction. Extract से बहुत simple aspect पे आपको चीजें मिल जाएंगी, so that isolation is little bit easy if you'll go with any sort of column chromatography or means other type of chromatography which are more sophisticated in the aspects. So what to do in that? If you have extract, say if you will, I'll take an example of very common plant like Vincarosia. Okay, it has the blend of different constituent. You have selected basically an hydroalcoholic extract. Considering the aspect like water will have major number of microbial growth, so you have taken alcohol also, and accessibility is there and various factors are there, and your chemical constituents you are getting to a major extract in that particular constituent. You have taken your preparation, you are extracted it, and you have got a dark green color extract. Now depending on the selection of the solvent and the chemical constituent, you'll make the chart of chemical constituent. Now you can select the solvents from non-polar to polar. Say if you'll start with pet ether, all lipid soluble constituents will go with the pet ether. Then you will take toluene, diethyl ether, and your ethanol, and then finally water. So what will happen? Your different chemical constituent will be separated with the mode of fractionation in different type of solvents. So your constituent or the chemical compound which you have received, your solvent which you have received, your fraction which you have received will be simplified to an extent. Okay, then after simplifying your material, you can move towards the further sophisticated instrument, because PET ether will take the lipid soluble molecule, they have low polarity. Ethyl acetate will take the medium polar, like monoglycosides are there, flavonoids are there, it will take with it. Now in butanol layer, if you'll take it, it will take somewhat with higher polarity or strong polarity. Compound which are water soluble, have strongest polarity, they will go with the water, like glycosides are one of them. So likewise, you will simplify your isolation procedure, you will get different fraction of your compound. Okay, so this is the solvent extraction method or solvent method by which you can simplify your isolation procedure. Okay, coming to the next procedure, that is precipitation. Forming a precipitate of some phytochemicals with specific reagent is another method which can simplify your thing, or which can help you to take your material out of a blend of material very easily by using the precipitation reaction. Okay, now coming to in precipitation, we have solvent precipitation method also. Okay, it say if you have the component mixture which have the mixed component solution, now what you will do, you will just add a specific solvent which is mutually soluble with the solution, so that you can separate your material with gradual precipitation. By changing the polarity, you can add different solvent and you can get your material by precipitating a different sort of different side with different sort of solvent, and you will get your solvent by fractional precipitation, which is a very important aspect, easily to be done in the lab scale. By gradual precipitation, you can solve, you can reduce the complexity of herbal compound. Herbal compounds are very complex in nature. Due to its complexity, we are going through number of procedure, we are going through number of separation technique, so that you can get an isolated material. This isolated material can be used to an extent of your utilization. Now what we are doing, we are employing advanced technique of artificial intelligence, advanced technique of separation, from starting from collection to end of the development of lead molecule. Okay, now it's been clear that we can go by precipitation. It can be reversible, irreversible precipitation is also there. Various type of specific reagents are also mentioned, like you can use Reynolds ammonium salt to precipitate, and after reacting with alkaloids, and they separate the alkaloids and non-alkaloids. One of the common example is there, Reynolds ammonium salt. Okay, now moving further, the next thing is salting. Dialysis, you can do it, you can salt out as per, you can separate the water soluble compound and other inorganic salt by salting out with sodium chloride is majorly used, sodium sulfate is used, magnesium salts are used, ferric sulfates, ferric salts are used for the separation. Like for an example, you can see that tetradyne is there, which is used, which is means like it is extracted from barberries plant, and you can use various salts to separate it. Okay, dialysis is also one of the method by which you can separate it. After salting out and dialysis come the fractional distillation. Now this particular method is the method of separating compound in any medium at different boiling points. Different boiling points are used by separating the compound by this particular method. They are categorized on the basis of atmospheric, vacuum method you can use, or molecular distillation it can be used. So majorly it goes with the volatile oils. You can use scavengers for separation of volatile oils. Because of the separation of volatile oils, you need to preserve the oil because they are volatile in the nature. Another example, you can also separate alkaloids from total alkaloids using this particular method, wherein different type of chemical constituents have different boiling points, and you can take it out and separate it down. I also mentioned the crystallization. It is also used. It is similar to the method of precipitation, wherein complex compound can be crystallized into pure substance and it can be separated. The very good example of crystallization is separation of caffeine from tea. What we do, we get the alkaloid, this caffeine alkaloid out of that mixture. The procedure involved various method of separation, which is classical method. If we'll start with the boiling of tea, we boil the tea. After boiling, first thing which we have incorporated is we are preparing decoction. After preparing the decoction, which I already told you in the first slide of the extraction, you go with the separation. Now you need to remove all the interfering things. I already told you, whenever you talk about the extract, it is a blend of chemical constituent. It is a complex material. It need to be resolved, it need to be simplified, it need to be taken to an extent for the compound of our interest. Okay, so what we were doing, we just boiled the tea and then we have separated the extract out of it. Now the first compound of interest or removal is that tannin. So what we do, we just put the lead acetate to it. Lead acetate precipitates the tannin. Here implies the classical isolation method of precipitation, wherein you can remove the tannins out of it. Means your solution is free from tannins. You can separate it, you can use it if it is your utilization parameter. Then afterwards, coloring matters are removed by using this charcoal. Charcoal remove or absorb all the coloring matter out of it. Here another separation is there. Then finally, by using solvent extraction method, solvent separation method, we are using chloroform, because this particular mureoxide alkaloid is highly soluble in the chloroform. So it's separated by the separation technique and you'll get the crystalline components which are alkaloids in nature. Okay, so these were all the classical approach which we have already discussed. Now moving towards the modern separation techniques, which are majorly used by various parameters of separation. So before starting any type of separation technology, most important aspect is to discuss about chromatography. We need to discuss about the chromatography because this particular method is highly important and accepted, or we can go it, all the other principles are dependent, all other separations of chemical constituents of natural product are dependent on chromatography. Now similarly, all the principles which we have discussed, selection of solvent, selection of material, implies with the chromatography also. Briefly, I'll just start with the adsorption chromatography. Adsorption chromatography is a chromatography which is based on the differences of adsorbing capacity of adsorbent. So it can be used, like you can go with the silica gel, alumina, out of which silica gel adsorption is majorly accepted and most suitable, because this particular chromatography have the major aspects of the separation. Sometimes we go for alumini also. This alumini adsorption is also there for specific compounds like alkaloids, steroids. Otherwise, in all sort of chromatography, starting from TLC to HPLC, we go for silica gel, depending on its size. Means the size it's vary because of the kind of sophisticated separation which we are seeing. Similarly, after adsorption, partition chromatography is one of the important aspect where which we can separate the component, and partition chromatography is majorly divided into normal chromatography, normal phase, and reverse phase chromatography. So if the polarity of stationary phase is stronger and mobile phase is normal, which is used to separate, then it is majorly a different type of chromatography. And if you have stationary phase very much polar and this mobile phase non-polar, it is reverse phase chromatography. So this particular chromatographic aspects are common, and column I have already halfly discussed with the classical isolation, where I have showed the precipitation on this. Column chromatography, we can go for wet column or dry column. Again, wet is highly preferred, so columns are available. Silica grade of column having mesh size from 5 to 60 mm is major, 60 to 120 is majorly accepted. Now we are taking it in the column with using silica gel with the solvent of your interest. Solvent like dissolve like, again the same principle lies over there, and then you will put it in the column without any sort of bubbles should not be there. Let the column settle, then you can put the compound of your interest. Again, if you will take fraction to it, it will be a simplified form. Rather than directly taking to the extract, you can go for solvent extraction. You use the solvent separation partition technique, and then you'll take your fraction. Based on the TLC, you select the fraction and you run the column. You'll get the different color bands at different RF, and you can select by collecting it by the column. Similarly, HPLC is a rapid method of separation and accepted throughout on the basis. It is again based on the older aspects of the column chromatography. Separation principle is same. Only the thing is that you'll get in pre-packed silica gel column is there with the filter. It's a column will have 5 to 20 mm size. High pressure is there with the high pressure pump. It is eluted inside so that it can be betterly separated and to and get in better aspect. Coming to the droplet liquid lipid separation technique, this is again a very important technique where the formation of droplets take place, which is required when mobile phase pass through the liquid stationary phase column, and droplet of the mobile phase get an intimate contact and forming a new partition over there, and by which the separation method is there. It basically is used for the solvent having the phytochemicals having the stronger polarity. Now these are the compounds or herbal compound which are majorly divided into various sectors like phytomedicines, nutraceuticals, cosmeceuticals, functional foods, dietary supplement, cosmetics, flavors, herbal botanicals, and these products have huge market. Therefore, we are moving herbal industries are moving towards standardization, development of lead products which can be majorly used for developing all sort of nutraceutical, cosmeceuticals, or cosmonutraceuticals are also coming in the market, which are getting a wide approach or wide acceptance due to its benefits. So now I can just show you this particular flow chart. All the brief history or brief discussion about all those topics are being explained over there only. Now we are going for drug discovery of medicinal plant. Madam, we have time? Pooja ma'am?
Ma'am, kindly cut it short. Actually, our new speaker is also going to join in a short time.
Therefore, I have asked. Okay. So basically, I'll just explain this particular procedure, and then we can move to the other aspect. So this particular chart will explain the development of development of herbal product, development of the constituent of herbal product by putting all the sectors over it, by separating the different aspect. Means you will identify the disorder, and finally you'll get the purified component by extracting and isolating the compound of your interest. Coming to the interesting part, drug discovery pathway, wherein we have discussed about the extraction and isolation. See, always before starting any type of extraction, any type of isolation, any type of drug discovery in phytochemistry, most important thing is the study of primary and secondary metabolites. Now whenever we talk about the primary and secondary metabolites, these compounds should be developed in all sort of herbal plant to an extent of their extraction. Here in the herbal industry, or incorporating pharma 4.0, AI is playing the important role, because by studying the intact plant, by providing the data for different type of temperature, pH, different type of collection time, we can develop, yeah, industry is developing a standardized parameter for collection of primary and secondary metabolites, because they are very complex material, and proper development in that particular stage is very important. Then coming to extraction, while extracting, they are incorporating process analytical technology, which is PAT, which is real time analysis of getting the product at right time, at right quantity, with right number of chemical constituent, and without degrading, because majorly the herbal plants are facing difficulty while extracting, to get a plant extract by degrading some sort of chemical constituents, which will not be degraded, incorporating different type of parameters like process analytical technique. Then you can screen the product, you can fractionize your product, and you can isolate the product of your interest with the structure identification by incorporating various advanced sophisticated technique, and you can lead to the drug discovery of any plant material. I already told you, drug discovery is a very time consuming procedure. It takes somewhere around 12 to 13 years to get a lead compound out of it. But if we talk about the herbal products, although they are complex, but they are important point of constituents to develop and to get the product of interest very easily, by using certain sort of complex parameters which can streamline your things, and you can get the product to be done structurally identified, because herbal means it is always a multiple constituent. Whenever you talk about the single to constituent is easy to evaluate, easy to get out of it, because its isolation involve use of various sophisticated product, and the product is streamlined, because you can develop the solvent of your interest, you can develop compound of your interest. But whenever you talk about the complex multiple product, say if it is a blend of multiple product, you require cumulative profiling of that particular drug using HPLC. If you need to get structure identification, you need to go for FTIR or different particular things. So these all techniques of separation are now integrated with artificial intelligence, so that you can streamline your product, you're starting from collection or starting of sowing. In the agriculture also, artificial intelligence is playing an important role for the selection of seed, for maintaining the optimum temperature, optimum parameters, which can create a greater library and database for better development of herbal products. So thank you very much.
Thank you so much, ma'am. Thanks a lot for such a wonderful talk. Actually, I was completely mesmerized with the topic that this drug discovery as well as the artificial intelligence along with the natural products. Thank you so much, ma'am. And I'm quite hopeful that our students and whoever is the audience, either the faculty members or the students, they will get benefited from this lecture. Thank you so much once again.
Thank you very much, man. I'm awaiting for the reply or response of sir.
Director,
So,
sir.
thank you very much, Dr. Sapna ji. So, you have delivered a very
The pleasure is
excellent
mine,
talk,
sir. No need of thank you, sir. Pleasure is mine,
and
sir.
you have given, and I have come to know that you have given, Dr. Pooja Madam, how many extraction techniques she is given. She is given
Pardon,
the
sir?
16 extraction technique. In a one slide, she has mentioned the 16 extraction techniques. So this is the excellent because I have also select some of the slide which are very very important because I know that nowadays people are taking the very very superficial knowledge and you have given very very vast technical and sound information to the you can say audience. So I hope that your this talk it will be the useful to those people who are the working in the herbal drug delivery system and even not they are the working the herbal those person they are the related to the extraction technique separation techniques they want to do the work on the herbal drug delivery system it will be the highly useful. So again I thankful to you for provide your precious time. I know that in those days academy is going on the very pattern admission all these things that going on. You are the one of the responsible person of your institute, so obviously time is very difficult. So three four things I want to mention over here. All slides are very sharp. Second, the formation of the slide, using of the words sentences they are the excellent, and your voice quality and sharpness is also excellent. So from my side 100 to 100. So thank you
Thank
very
you
much,
very much,
madam.
sir. Thank you, sir. Thank you, ma'am. Thank you very much.
Bye bye.
Okay, ma'am. Thank you.